Hello, this is Monika Sommerhalter. Welcome to the Chemistry 443 video series. In this video, I'll show you how to conduct the Bradford assay, which is used to determine protein concentrations. In this video, we'll first have a quick look at the Bradford reagent, followed by the assay principle, and then I'll walk you step by step through the procedure. The Bradford reagent is based on the dye Comassi Brilliant Blue. It is dissolved in methanol and phosphoric acid, and as you know from your safety assignment, those two chemicals are very hazardous, so please dispose all your reagents in the acidic organic waste. Here is the structure of Comassi Brilliant Blue. It has several phenyl rings and also two sulfonic acid groups. Comassi Brilliant Blue forms complexes with proteins, and it favors proteins that have many aromatic amino acids and amino acids with positively charged side chains. As Comassi Blue forms complexes with proteins, its absorption spectrum changes, a bright blue color forms, and the absorbance at 595 nanometer increases, and that's the reason why we choose this wavelength to monitor the protein content. Here you see two test tubes, one test tube filled with Bradford reagent and a little bit of water, and the other one with Bradford reagent and a protein standard called BSA, or bovine serum albumin. You can start your experiment by diluting your sample. Here's the formula for your sample dilutions. Start with a P1000 pipette, set the volume to 900 microliters, and then deliver these 900 microliters into a pre-labeled microcentrifuge tube. To set the volume, first open the lock, then turn the knob to the desired volume, but never turn a pipette outside its specified volume range, and then finally close the lock again. It's a good idea to pre-wet a pipette tip for more reliable aspiration. Don't immerse your pipette tip too deeply. Here I'm preparing two different types of samples, one for banana pulp and one for banana peel, but in your experiment we'll be focusing on the banana pulp sample. Next, I set the pipette to 800 microliter. I can still use the same pipette tip because so far I've only been working with the distilled water, and I don't need to pre-wet the pipette tip while delivering the very same type of solution with the same pipette tip. Then I switch to a P200 pipette, and I'm thoroughly mixing the crude extract so that I get a homogeneous solution before I take out a small portion. Don't immerse your pipette tip too deeply into the crude extract, just a few microliter, and directly pipette into the diluent, in this case into the water solution. So the second volume is set to 200 microliter. I use the fresh pipette tip, and I'm mixing by pipetting up and down. Since mixing is so important, I'm also inverting the tube several times. Next, you can move on to the bovine serum albumin dilution series. The dilutions are calculated with the formula C1V1 equals C2V2. Here you see one example for one of these dilutions. We'll be making a total of 6 dilutions, and then there's also the water sample. Here you see the bovine serum albumin standard. It's provided in the form of an ampule with a very exact 2.00 milligram per milliliter solution. To break, or concentration, to break open the ampule, I'm wrapping it in a paper towel to break open the neck, and then I'm transferring the solution into a labeled microcentrifuge tube before discarding the ampule into the glass waste. This is the P1000 pipette, and I'm starting with the larger volume. I'm starting with the diluent, and in this case we use water. Again, I'm pre-wetting the pipette tip. It's a good idea to touch the side of the container when you deliver the solution, and all the microcentrifuge tubes were labeled. The 0.1 stands for the 0.1 milligram per milliliter bovine serum albumin that I want to have in this particular tube. So the video runs at faster speed because it's basically always the same process, delivering different volumes of water, distilled water. Whenever you change the volume, open and close the lock, and now we move on to the bovine serum albumin standard. I'm pre-wetting the pipette tip 3 times. And then I'm pipetting directly into the solution, into the larger volume of the water. Pipetting up and down to mix the bovine serum albumin into the water. And I'm even flicking the tube a few times. You could also invert the tube to have a thoroughly mixed standard. Now at faster speed, we'll be working our way through all the dilutions of your bovine serum albumin standard. Always check the volume that you set your pipette to. To deliver a solution, you press the piston all the way down. Now you need to change pipette tips for every delivery. And then here in this part, I'd like to emphasize the position of the piston. So there's a first point of resistance, which defines the volume. So you press down to that first point of resistance before you immerse the tip into the solution. So just a few millimeters under the surface. Then you slowly release the pressure onto the piston or on the piston with your thumb. So this is the motion to pipette up and down to pre-wet the pipette tip. Then I'm aspiring or sucking up the solution one last time from that first point of resistance, releasing the thumb very slowly. And then when I pipette into the water, then the pipette tip is not really all the way in the solution, just a few millimeters, but then I press all the way down with my thumb to the last point of resistance to fully eject all the solution that is in my pipette tip. You can mix by pipetting up and down, but after closing the tube and flicking or inverting the tube, the solution should be thoroughly mixed. And here is the last dilution of the bovine serum albumin standard. To conduct the Bradford assay, you need to work with the Bradford reagent. So don't forget, it is very corrosive. It is also harmful. You will have your safety goggles on and gloves, and since it's Corona time, also your mask. So please use a 5 milliliter volumetric pipette to distribute the Bradford reagent into several test tubes. Here I pre-labeled the test tubes to avoid confusion. We start to add 0.1 milliliter or 100 microliter of either standard or sample to these pre-filled tubes that already contain the 5 milliliter Bradford reagent. And as always, mixing is so important that I pre-cut some parafilm that I'm always wrapping tightly around the top of the tube so I can mix thoroughly by inversion. So here you can see how the blue color doesn't reach all the way down. I actually really have to invert the tube. Here is this comparison between the two extremes, just 0.1 milliliter water versus 0.1 milliliter of the highest bovine serum albumin standard. And then one by one, right, the tubes are prepared. I'm working my way through all the standards. And in between, I always have to use a fresh pipette tip. So here is a lineup of all the tubes that I prepared with the bovine serum albumin standard. You should see that transgression from brown to blue. Then the banana pulp extract is added, so 0.1 milliliter of the undiluted banana extract. And I'm getting a really bright blue color, at least, if not more blue in tint than the highest standard. Next, I'm working with the 5-fold diluted banana pulp extract. And it seems to me that this bluish tint kind of falls in the middle of the lineup of the standards. The 10-fold diluted crude banana pulp extract is treated exactly the same way. Actually, the standards as well as your samples, it's always the same procedure to be able to make these comparisons. Here is a lineup between the 3 different dilutions: 10-fold, 5-fold, and undiluted. I decided to use the 5-fold dilution because it fell in the middle of the standard curve, and I'm making new dilutions. So 800 microliter of distilled water and 200 microliter of the banana pulp extract. I'm starting at this stage at really preparing the sample dilutions again. And here is my final lineup. So the banana peel will not be part of your experiment. And as you probably saw, there's not much protein content in that sample. Next, you set up your nanodrop spectrophotometer. You need a USB cable to connect the nanodrop with the USB inlet of the laptop. And you also need a power cable. Without power, the nanodrop 2000C will not work at all, so that can be a reason for a failed measurement. We select the UV Vis option of the nanodrop software. On your own, you can explore the Bradford Assay. So don't forget to press Use Cuvette, else the instrument will measure at the pestle. Enter the proper wavelength, and then press blank to start your first measurement. To transfer your samples and standards with the Bradford solution, I simply poured. You could also use a glass pipette or another pipette. And I'm wiping the outside of the cuvettes with a Kim wipe. It's important to fill the cuvettes with at least 2 milliliter sample. You don't have to fill them all the way before you insert the cuvette into the slot of the nanodrop spectrophotometer. The light path is indicated with the arrow. So if you have cuvettes that have a milky or opaque side, make sure you put the clear sides so that the arrow goes through the clear sides of your cuvette. To start the measurement, put the arm of the spectrophotometer down, and then you simply press measure. You'll be asked to enter a file name, and then one by one insert your cuvettes You can provide sample names, but please keep track of all the absorbance readings in your lab notebook.